Hello, everyone. This is the PowerPoint for Chapter 8, and I'm going to do all of it all together in one video so it's not confusing. And then you can skip along to different parts if you're in a different section. And I also posted the PowerPoint. It's a student version. I'm also going to go through the normal paddle questions that we do at the end of this. And that's not on your PowerPoint, but I thought we could go through that, and I won't take a grade, but if that's something that you wanted to review that you would find helpful, I'll add that to the end of the video. So this will be a pretty long video. We'll see if we get through it. So first of all, I wanted to start with talking about the COVID-19 disease and the SARS-CoV-2 virus, because a lot of what we do can be centered around that. I know you guys are really interested in learning more about it, and I'm also really interested in that. So I thought we could focus this chapter around that, and I added questions to each of your worksheets on SARS-CoV-2, which is the virus name. And then the end part of this whole of Chapter 8, your test, will be just an essay talking about what you learned. And I put a prompt, but you can talk about other things too that can be kind of open-ended. So I wanted to start explaining some of what we're looking at with SARS-CoV-2, which is this virus that causes COVID-19. This is what it looks like. I'm sure you've seen this picture all over. This is a model of it, probably taken from a scanning electron microscope and false colored. And it's beautiful. It's a beautiful virus. And we'll kind of dig into what we're seeing here. I did not, now that I'm thinking about it, I didn't put a picture of what's inside of this virus, but if you were to crack this open, here on the outside, you just have protein. It's just a protein shell. And then on the inside is just RNA. That's it. This is not a cell. It's not alive, technically, even though it is causing a pandemic. It's just a protein coat and an RNA inside instead of DNA. So that's a little bit of a difference between cells and viruses. And remember, this is much smaller than a cell too. So it's crazy that this much chaos can be caused from something that's not even living, technically. So I wanted to show you the mechanism of how this virus actually infects. And this is all really new stuff. I haven't had a lot of time studying it. I'm learning it alongside of you guys. So I don't understand all these mechanisms, but I wanted to show you, this is just a typical coronavirus. SARS and MERS are also coronaviruses. This was from a study from 2014, I think. And it shows like the process of how a virus, these coronatype viruses can attach to the cell. Let me get where's my special mouse thing? Oh, I don't see it now. Okay, well, that's weird. Hopefully you can see my mouse. And I can do some drawing, I guess. So here is the virus. It's going to attach to the cell. And then it actually moves into the cell. I'll show you a picture of that in a second. It becomes uncoated. So the outside protein opens up and kind of dissolves. And then it gets into the RNA. And all of these steps happen, but basically that RNA is going to become translated into proteins. And then the cell is going to be able to make more virus. So you can see here it's actually building the virus. The cell basically becomes a virus factory. And then it puts it into these little vesicles, which are made of cell membrane, and then it shoots it out through exocytosis, which we learned about before. Okay. How do I get to the next slide? Next slide. All right. Here's another more simplified version of it. We see here that SARS attaches to this receptor, and then it enters the cell. So this blue part here is the cell. And then the shell, the protein shell of the virus kind of dissolves, and it releases the RNA, which is the genetic information. And then the cell actually does translation, which is a term we'll learn about in this chapter. And it actually starts to make new cell parts. You can see here the new cell parts are being made, just like a cell would make any protein. And then it's going to start pushing it out. And my understanding is that this cell doesn't die necessarily. It can keep making virus. All of those things are really important to know when we're studying this, this infection. And this is from 2019. And this may not be SARS-CoV-2. This could be another coronavirus, but in this case, they're labeling it as SARS-CoV-2. So this very recent study here is saying this is how they replicate. And then here's an even more simplified version. So the coronavirus comes into the cell, and then the coat dissolves, and you have the RNA here, which is in the middle. So you just have a protein coat and then RNA in the middle. And then if we come down here, the cell starts to create coronavirus parts through translation, and then it'll do exocytosis and send them out. So that's kind of an overview about COVID-19 and SARS-CoV-2. And as we're studying DNA and how it works in the cell, I want you to keep that in mind, and we can try to apply that to SARS-CoV-2 and this disease. So here's some information that I wanted to tell you. I did do that other video, but I wanted to give you another overview on why this is such a big deal, why this is considered a pandemic, and why we're shutting down schools all over the world. I know teachers in other countries too that I've worked with, and they're also shut down. Some of them, like in South Korea, they're thinking about opening, but as a whole, this is really affecting the whole planet, which is wild. And I've heard some people compare this to like the flu. Why is this different from the flu? So here's why. I'll give you some information on why this is such a bad virus and why we're so worried. And so I couldn't get these to come in one at a time. I don't know why. So here they are. We'll go start the first one. Right now, currently, according to my calculations, and this is changing every hour, but right now the global fatality rate is about 4%. So 4% of people are dying. That means if 100 people get it, 4 people are going to die. If 100 people in Baker get it, out of that, 4 people are going to die. And then on top of that, probably 4 to 8% on top of that are going to be hospitalized. So you're looking at about a 20% chunk. 20 people out of 100 are going to be severely ill. And maybe 15%, maybe 3/4 of those would then have to be hospitalized in intensive care. And that can also get our medical workers sick too, which has happened, and some have died even in the US. So that's really concerning. We can't really handle that many people in the hospital at once. We already have a lot of people in the hospital for other reasons. And then what we're seeing is that this fatality rate of 4% is at least 30 to 100 times higher than the flu. So the flu is hard to calculate, but it's probably between 0.5 and 0.05 and 0.1% fatality. This is way higher than that. So those two reasons, that first reason, sorry, is one of the reasons that we're concerned. And then another issue that makes it different from the flu is that it has a very long incubation period, 3 to 14 days. So somebody can have the virus and be shedding it for 2 weeks before they know it. And this actually happened in a cluster in South Korea. I think patient 31 probably infected 1,000 people before she knew she had it. And she made some bad choices too, but that can happen. And that's a really, that coupled with the 4% fatality rate is really a nasty situation for a virus. It also infects 2 to 3 people, which is higher than the flu. The flu is about 1.5. And so each person can affect 2 or 3 or more, 1,000 in her case. And the other issue is that people without symptoms can be really contagious. And so that's concerning too, because people don't even know they're sick. Only 20% of people become very sick. The other 80% are not so sick. That's also a problem. That's what makes this different from SARS, which was an epidemic, a really nasty one, because with SARS and MERS, most of the people got super sick. In this one, 80% of the people, 4 out of 5, are fine, and they're walking around spreading the disease and not knowing it. And so that's really hard to contain. It does spread really easily too. And the other concern that we have and why we're shutting things down is because here in the US, the number of infections have been doubling every 3 days. So that's exponential growth. And if we keep doubling at that rate, then in a month we'll have 1 million cases, or millions. Probably, I think I calculated 1.5 at our current rate, 1.5 million. And then when we get to numbers that big and it keeps doubling every 3 days, if we're at 1.5 million, then in 3 days it'll be 3 million. In 3 days it'll be 6 million, and you can keep going from there. So that's why we're concerned. That's why we're shutting schools down. We're trying to get around this, and we're trying to do that flattening the curve, spreading out the infection so that our medical staff can handle it. Okay, so let's shift gears into chapter 8, and we'll try to tie this together with what's happening in our world as much as possible. And so chapter 8 is on DNA and how DNA works. Now, the difference is that COVID-19 doesn't have, or SARS-CoV-2, the virus, doesn't have DNA in it. It has RNA, which is a little bit different. And we will still look at these processes with DNA, but just know that the process with SARS-CoV-2 is a little different. And what we'll start with is how DNA was actually discovered. So to begin with, DNA they didn't think that DNA was the genetic material that was being passed on when Mendel was doing his experiments with pea plants. They knew something was being passed on between different generations, but they didn't think it was DNA. They thought it was proteins, because DNA is like super simple. There's not a lot to it. So they didn't think that could be it. So there's some famous experiments I wanted to tell you about how they actually discovered that DNA was the stuff. They knew about it, but it was the stuff that was transforming things that was being inherited. So the first one was a guy named Griffith, and he was working with mice. And he was studying some bacteria that cause pneumonia. So he took this live bacteria, and it was an S form, so it was smooth. He injected it into this mouse, and it died, probably of pneumonia. Then he took some R strain, which was the rough strain. So this is just a different strain of this pneumonia bacteria, injected in the mouse, didn't kill it. Then he heat killed the deadly S strain. So he flamed it. He stuck it in a flame, killed the bacteria. He injected the mouse, it lived. Then he took the R strain that was alive, the heat killed S strain. He mixed them together, injected the mouse, and it died. So something happened here in this one. This is weird. This would have blown your socks off to think that some dead harmful bacteria and some live fine bacteria can kill you, when the two separate here don't kill the mouse. So these two scenarios don't kill the mouse, and this scenario does. So what he concluded is that something was being passed from the dead bacteria into the live bacteria and transforming it. So he called that a transforming material or transforming principle. I'm going to take a drink. Okay, so that was the first one. Very interesting. Avery picked it up. I don't remember how many years later. It's in your book. Avery was the next guy. He was like, this is neat. So he went on and he designed 3 experiments. First, he kind of did similar studies to Griffith, but he looked at what was being transformed from those 2 bacteria, and he did qualitative tests, and he found that DNA was present, and then he did chemical tests and said, yeah, DNA is present, and then he did these enzyme tests where he put enzymes that only break down DNA, and it stopped the transformation. It stopped that transfer from the dead harmful bacteria, the S, to the safe R strain. So he's like, okay, these 3 tests are saying the same thing. We're finding that this is probably DNA. But at that point, scientists are skeptical, which is good. It's fine. So they didn't really believe. They were like, no, we still think it's protein. So they went on. And then I know this one was in 1952. So this was quite a while after. I think about 20 years after Avery. And these 2, they wanted to see if it was, they wanted to confirm it. And I want you to realize 50 years is not that long ago. My dad, or 1952, my dad was 5 then. So in my mind, this was not very long ago that we were figuring this out. So they actually used viruses. And one of the questions on your worksheet is how does this particular study relate to COVID-19 and SARS-CoV-2? So I wanted you to see that they were studying viruses also. And in this case, these viruses had DNA and SARS-CoV-2 has RNA. So it's a slight difference, but still relevant. They were using viruses that infect bacteria, which are called phages. And Some of you have worked with phages if you work with the Montana Tech people. So this is a phage. It's landing on the surface of a cell. And the phage, just like the SARS-CoV-2, is only made of a protein coat and DNA inside. And so they tagged this. They tagged the DNA with radioactive phosphorus because DNA has phosphorus and protein doesn't. And then they tagged the protein coat with radioactive sulfur. So they just like marked it so they could track it. They marked this DNA and they marked the coat with radioactive. And what they found was when that, and your book has a better picture of this, so look in your book for this part. When the DNA, when the bacteriophage infected the cell, what they found inside was radioactive phosphorus, which is what they tagged the DNA with. So then they said, yes, the phage is injecting DNA into the cell, and then the cell starts making more phages. So lots of similarities to COVID-19 here. But that's how that was like, this is like the hallmark study of how they were like, finally, okay, it is DNA and not protein. All right, so that's section 1, and you can do the worksheet for that. Section 2 is on the structure of DNA. And so that's basically what it looks like. And there's also an interesting story about how the structure of DNA was figured out, because it wasn't easy. It was a tough thing, and some bad things happened. So here's the structure. And these are things that you should write down in your notebook. You can pause the video or use your PowerPoint too. So DNA is made of nucleotides. Remember my 3D model that we have in the classroom that we're not in. And this is just one little piece of it. And so in this DNA molecule, we have a phosphate, we have this sugar group, and then we have this nitrogen containing base. So you have these 3 parts listed here: phosphate, sugar, and base. This base is where the information is. That's where the business end is. And I've heard a stat where if we had a gram of DNA, we could store enough information for 70 computers. No. I think it's 70 terabytes. I have, let me hold this up. I have this little hard drive. If you've seen these, I got it for Christmas, and it stores 1 TB, 1 terabyte of data. I have like 7 computers on here, and it's like 1/10 full. You would be able to put 7 of these in 1 gram of DNA. So it's a tremendous way to store information. And this is where it's stored, this part right here, the base. And so you can have 4 different bases. So that's that end part. They can be 4 different things. They can be a T, which stands for thymine. They can be a C, which stands for cytosine. They can be an A, which stands for adenine, or a G, which stands for guanine. And they have special name, like categories. So do you notice that this one has 2 parts? It has 2 rings, a pentagon and a hexagon. Same with this one. And then these only have 1 ring. And these that have 1 ring are called pyrimidines. And then if they have 2 rings, they're called purines. And there's a way to remember this. So I'll do it down here. This is one way that I like. It says cut the pie. P-Y. I'm spelling it weird. And then I'm going to draw a hexagon, which kind of looks like a pie. So what I wrote here was a way to remember which ones are pyrimidines or which ones are the single ring. So we have the C is the single ring. There's your C. The T is a single ring. There's your T. These 2 are the single rings. Cut the pie, pyrimidine. But U is also a pyrimidine, and that's in RNA. So we'll learn about U later, but it's another pyrimidine. And then the other 2, A and G, are purines. The other thing that we know from these is that a pyrimidine always bonds with a purine, and that probably doesn't make a lot of sense right now. So let me flip to show you a picture of what I mean. Let me just keep going with the story. So Watson and Crick are two people that are really famous for helping to discover the structure of DNA and what it actually looked like. They were really confused and also egotistical and kind of a lot jerks. And they were trying to figure this out, but they needed some help. And there was another person working on it. And oops, where did it go? Her name was Rosalind Franklin. So there's her name there. And then this other guy, Erwin Chargaff, he was working independently. He was looking at which bases bond with each other. So he found that A's bonds with T's, C's bond with G's. So Rosalind Franklin was working on X-ray crystallography. So she was like drying DNA molecules and then doing this really difficult X-ray work to get pictures of them. And she was really good at it, and it was really difficult. And so Watson and Crick, the two other guys, needed her help, but they were huge jerks to her. And she was able to obtain this famous photograph called Photo 51. And this actually helped them to see what the model should look like. And they wouldn't have been able to figure it out without that. So they actually, the way that the story goes, and they actually wrote a book about it, they went into her office and they found this photo when she wasn't there, and they sketched it on a piece of newspaper without her permission. And then they went back and made the model, and then they asked her to come and look at it, and she was like, yeah, that actually does look consistent to my picture. And then they like published on it, and they got the credit, and they got a Nobel Prize. And then she went to another university and ended up dying there of cancer because she had been exposed to so many x-rays. So that's kind of the story behind the discovery, and it was messy, and they were really unethical. And it's too bad it went that way because it could have gone better. They could have collaborated together on it. So this is what it looks like. These rungs right here, it's like a twisted ladder, right? And these rungs are those bases, those A's, T's, C's, and G's, right? Those purines and pyrimidines. These are the bases. And they bond with each other in a certain way. So you can see here that the reds always bond with the greens, and the yellows always bond with the purples. So they always bond that way. And it's like a twisted ladder, right? And Chargaff is the one that figured out that those bond together. And then the other, well, let me put this rule here, and hopefully you're writing these down, but A's bond with T's and C's bond with G's. And here we have, this is a pyrimidine single ring bonding with a purine double ring. And then here we have a pyrimidine single ring bonding with a purine double ring. So the single rings always bond with a double ring. So here, if we look at that, I'm going too far. Okay, the single rings are always going to bond with a double ring. A's bond with T's, C's bond with G's. Okay, so that's Chargaff's rule. Okay, and we have all of that. So that's what it looks like. And there we have that information. Single rings always bond with double rings, and that keeps the width the same. If you had like two double rings, it would be too wide. And if you had two single rings, it would be too narrow. So that's why we see that. Computer. The backbone here, the phosphate and sugar, those are covalent bonds. They're kind of like hard bonds. So covalent bonds are like a piece of paper. When I rip this paper, that's a covalent bond. And then inside here, between these nucleotides, those are hydrogen bonds. Those are really weak. Hydrogen bonds are the bonds that I'm making with this piece of paper, or that you might be making with, like, if you're holding your phone or holding a pencil, those are hydrogen bonds. They're really weak, and that's important. They have to be weak because this right here has to unzip to get to the information, to be able to like replicate the DNA or code it. So that's why these bonds are weak, and that's why that's important. All right, we're moving on to 8.3, which is how DNA actually replicates. All right, so this is something we've seen before. So this is our cell cycle. And remember, in these phases of interphase, the cell's growing, doing its job. Then we hop over here to synthesis. And hopefully you're saying this with me. Wish I could see you guys. That's where the cell is replicating its DNA. So it's copying its DNA to get ready to copy itself. And that's where we're focusing. We're focusing on the synthesis phase and how the cell actually replicates its DNA. So that's where we're at. The DNA is a template, and then you have all these little busy helpers doing this work, and they're called enzymes. So they're all coming in, helping to replicate this DNA. They unzip the double helix, and then they come in and bring in new nucleotides that you ate when you ate strawberries or something. So here's what that kind of looks like. We're unzipping these hydrogen bonds. The covalent bonds are staying the same because they're harder to rip, but we're able to unzip, and then we bring in new pieces. So you can see these little orange new pieces that are coming in and bonding. And they're doing it kind of opposite. The DNA molecule is anti-parallel, so it goes like this, opposite directions. So you're gonna have like DNA being made over here and over here. It's not gonna be on the same side because they're opposites. All right, so here's some of the enzymes that are doing the work, and you'll have some questions on these. This is DNA polymerase, and it's doing the work of bringing in the new nucleotides and bonding them. Polymer means like a, what's the best way to describe it? A polymer is like a whole bunch of the same units added together, like a beaded necklace. So like nylon string is a polymer because you just keep putting the same molecules together and building like a nylon string. And so polymerase makes sense because you're putting all these little pieces together in a chain. So polymerase is bringing in the nucleotides and hydrogen bonding them, and then it's making these covalent bonds with the backbone. And so you can see that we're unzipping and we're getting 2 new strands from this eventually. So here is that after it finishes, and we personally have 3 billion of these rungs. So you can tell that this would take a long time since it does, we're doing like a whole bunch of different spots at once. We're doing it in different places on different chromosomes. So you get 2 new molecules, and this is called semi-conservative because you have 1 old strand and 1 new strand. So that is semi-conservative. Okay, so this replication can happen really fast in lots of different places. So you're doing all these, you have these little forks, they're called replication forks. You're doing them a whole bunch of times all at once, and you're able to make 2 strands really quickly. And then the DNA polymerase is doing this work, and it's correcting mistakes too, which is nice. Another enzyme that I don't know why I don't have on here, but it should be in your book. The enzyme that actually unzips right here. So let me draw it for you. Okay, so right here you have an enzyme that is called, I can do text, called helicase. And that is the enzyme that unzips the helix. This is called a double helix, and it unzips it. And then another enzyme that you have right here is called DNA ligase, and that's what glues the like here, the backbone back together. So that kind of ligase like means like glue, so it's gluing that back together. Okay, I'm gonna clear these. All right, so that's section 3. We're cruising right along. Now, section 4, we're totally switching gears. What we were just looking at is how the cell is going to replicate its DNA to replicate new cells. And now we're looking at how the cell is going to actually make protein. So this is more into how like SARS-CoV-2 would be replicated in a cell, like how your cell would make more viruses. But some of it's a little different because this step is how we make RNA, but with SARS-CoV-2, the genome is already RNA. So we don't actually need this transcription step. We can skip right to making protein, which also might be why this is such a virulent little bugger, because we don't even have to do transcription on it. It's possible, I don't know this for sure, but it's possible that viruses that have DNA as their genome are going to take a little longer to replicate because they have to go through this step. So that's a maybe. So just things to think about. And you can look that up and correct me for sure. All right, so what we're doing here is we're converting the DNA, the gene, into RNA. That's our first step when we're going to make a protein. Okay, so this is called the central dogma of biology, and I'm going to take a sip of coffee and let you look that over, see where we're at and what we're doing in each space. Okay, so we are in the nucleus. We're in a eukaryotic cell, yes? And in here, we can replicate our DNA. So that's what that part's saying. That's not really part of this. That we're doing that when we're going to do mitosis. That doesn't matter. But we are also going to change DNA into RNA. So we're doing transcription, and we are transcribing DNA into RNA. And we're doing that in the nucleus. And there's some things that happen to the RNA before it can go out. And then once that happens, it gets edited. And then it leaves, this is kind of a misleading picture. The RNA is going to leave the nucleus and go out here into the cytoplasm. And that's where it's going to make protein. And so if we're looking at it with SARS-CoV-2, we're just doing these two steps. We're doing RNA from the SARS-CoV-2, and it's being turned into protein and being constructed into a virus. Nasty. Okay, so these are the three processes, but this one shouldn't really be included. So replication is just the DNA replicating, it's going to do mitosis. That's different. And then these two, transcription and translation, are these processes here. So let me type these so we have a clear view of this. When we are changing DNA into RNA, we call that transcription. And then when we are changing RNA into protein, we call that translation. We got to keep those words straight. Also, transcription happens in the nucleus, and translation in eukaryotes happens in the cytoplasm on ribosomes. I don't know if you can see that over there. Hopefully you can see ribosomes all the way. Okay, so that's where we are, and that's what we're looking at. And there they are. So that's replication, transcription, and translation. Oh, and one last thing. The reason why we do RNA, this is our little messenger. The reason for that is because DNA cannot leave the nucleus. If it does, it'll get destroyed. So instead, it needs to send out a little messenger to do its dirty work, and the RNA will get destroyed. But we just need to not destroy the DNA. That's important. You need that. Okay. So here are three ways that RNA and DNA are different. First of all, RNA has a ribose sugar. DNA is called deoxyribose. I probably didn't tell you those terms all the way, so let me write that here. DNA equals deoxyribonucleic acid. It is a mouthful. There's DNA. And then RNA is just ribonucleic acid. There's RNA. So it's just missing that deoxy. That actually just means that DNA is missing an oxygen and RNA has the oxygen. It just has a longer name. So that's one difference. It has a different sugar. Remember, DNA has three parts. It has a phosphate sugar, and the nucleic acid. And RNA just has a different sugar. And then we also have uracils instead of thymines. These are both pyrimidines. Remember, cut the pie. But it's just a little bit different. It's a tiny difference in the structure. And then the big difference is that RNA is single-stranded and DNA is double-stranded. So RNA, it's basically like you cut a ladder in half and you just get half of the rungs. Let me clear. Okay. And so here's what happens. This is our big picture here. The DNA is basically copied by a strand of RNA, and it just makes a little piece of RNA for a certain gene. And here's what it looks like. You have this big transcription complex here, and it unzips it just like it would replication, and then it's going to bring in little pieces of RNA to make a little segment of RNA. So we'll see if I have a better picture. There's your picture of RNA that's being made. And it does a whole gene. So this is like 20,000 of these little rungs, like a lot. And then once that piece is taken, the DNA is going to zip back up and just be fine. It'll just go right back to normal. Okay, so there's what it looks like. That whole complex, that whole transcription complex just dissolves. It'll get used again later. And then there's your RNA. That RNA is going to get edited, and then it's going to leave the nucleus to go get translated into protein. And the reason why we don't let the DNA leave is because it'll get destroyed. And there's 3 types of RNA. What we've been talking about is messenger RNA, and that's just the little guy that carries the message to go make a protein. But there's also rRNA. That's what ribosomes are made of. So they're going to be made from the same process. And then tRNA, we will meet in section 5, and that's going to bring amino acids into the cytoplasm to make proteins. So those are your 3 types of RNA. Okay, this is what it looks like really happening. This like chunk right here would be one gene that's being transcribed. And then all these little noodles are like different RNA that's being made. You can make a whole bunch at once, and you can make tons from that one gene, and then it's going to go out, leave the nucleus, and make a protein. So you can see, isn't this beautiful? It's happening like in all, these are all genes. That's a gene, that's a gene, that's a gene. They're all making like this whole, like the main trunk, that main vein, those are the genes, and then you're making RNA. Okay. So now we're on to section 5, which is translation. So now we take this mRNA, this messenger RNA, it's going to leave the nucleus and go out into the cytoplasm onto a ribosome to make a protein. So we're going to make, let's go make SARS-CoV-2. Let's go make the virus that causes COVID-19. That's what we're looking at here. So to do that, this is a piece of RNA, and it has these bases. So remember that RNA has U's instead of T's. So we have like AUG, CUU, and it's only half the sequence. It's like a ladder cut in half. And this codon here has 3 nucleotides. So let me move on. So, okay, I guess I didn't explain that very well. A codon is when you have 3 nucleotides in a sequence, and that's going to code for 1 amino acid. So here we have this codon, AUG. It's going to code for MET, which is methionine. And then these 3, CUU, are going to code for leucine, which is another amino acid. You've probably never heard of those. You might have heard of some other ones. This is a chart that should be in your book, and it's how we code for proteins. And I didn't ask you a lot of questions about this. If you're planning on taking like AP Bio or anatomy and you want to know more about this, this is important stuff and we're kind of losing out. So I can give you like more information and more practice on this. Just let me know if you want more. So in this chart, we're able to look at the mRNA codon sequence and figure out what amino acid it codes for. So these weird names like phenylalanine, leucine, isoleucine, methionine, those are the amino acids. We have 20 amino acids that make up all the proteins in our whole bodies. And this is true of most organisms on Earth. Most have the same 20. There's some variations of that. So all these amino acids make up the proteins that we have. And if you see like something different than this on a protein shake, sometimes they list their amino acids and they're not one of these. That's because it's not actually an amino acid that we use. We're able to make 12 of these on our own, and we have to eat the other 8 on this list. And so that's why it's important that we're eating like meat or sources of protein like beans, peanuts, things like that, because we can't make all of them. And so you can see that some of these codons code for the same amino acid. Leucine has 6 different codons. Look at those that will code for it. And some of them only have 1. Methionine only has 1. This codon right here is the start codon. You have to have an AUG to start coding. And there are 3 stop codons here, UGA, And then here are the other two, UAA and UAG. Those are, so when you're looking at an mRNA sequence, it's going to start with AUG and it's going to end with one of those 3 stop codons. And that's how the ribosome knows where it's supposed to start and stop. And if there's a mutation in those, if something changed, bad things can happen. Or if something changes to a stop codon too early, a mutation there, it's going to stop that sequence and it's not going to make the protein. So that's how we use this chart. And here's an example of how you would use it. If this is your codon here, and you're going by 3s, it's by triplets. These are your amino acids. And then let's say that you were to shift over and start one over. So instead of starting at CGA, you're going to start at GAU. You're going to make totally different amino acids. This is a way where we can pack tons of data into a small space. You can actually, this is mind-boggling to me, but you can code a whole bunch of different proteins with the same reading frame, the same thing. Imagine if you could do that with a computer program. And in the same computer program, if you could code like many different files using the same data. So that's pretty mind-blowing, and that does happen depending on your reading frame. But no matter what animal or virus we're looking at, this chart basically holds true in all living things, which is pretty interesting. Okay, this is how it works. This is your tRNA, which is a type of RNA, this blue thing right here, and we see that sometimes it's shaped like kind of a T shape. So, and then we have this little anticodon on the bottom, and this matches the mRNA. So that's actually gonna bond using hydrogen bonds to the mRNA, and it's gonna be like complementary. So like the A is gonna bond with the U, the C is gonna bond with the G, etc. On the end of this little guy, he has his own amino acid that matches that anticodon. So each of them have a different anticodon with different letters, and it's going to match that amino acid based on that chart, based on this chart. Okay, so you have lots of different tRNAs. They're going to have their matching thing at the bottom, and they're going to come in to save the day. They're going to bond with the mRNA and drop off an amino acid. So here's what a ribosome looks like. This is where it's happening. And we have this large, it's like a little hamburger. You have the large unit up above and the small unit down below. And the mRNA is going to zip right through that. And the tRNA is going to come in and bond with it and drop off amino acids. And this is probably kind of hard to picture. But here is a good picture of it that I like. So here's the nucleus. The mRNA is quite long. It's coming out of a nuclear pore. It's already been edited. And then it comes and lands on a ribosome. So this is a ribosome, the little cheeseburger. The tRNA is going to fly in, land, and here's where we have the anticodon bonding with the codon, and then it's going to drop off an amino acid. And eventually, you can see here in the background, you're building these amino acids that become quite long proteins. Okay, so here's another view of that again, kind of a zoom in here. Here, the green, we have the ribosome, and then the red thing is the mRNA. And these are going to zoom in, bond, and drop off amino acids. And then eventually they're going to keep going. Once you get like 20,000, no big deal, it's going to make a protein. So this is a long process, happens really fast, pretty amazing. Then that little empty tRNA, ooh, where'd we go? Where'd he go? Where's the empty one? Okay, like this little guy, he's empty. He dropped off his amino acid, and he's going to float away, grab another amino acid that matches him, and come back and keep coding. And you can see the methionine is the first one. That's the start codon. So that's the very first one that has to be made. If that's missing, it won't be made. So there it is. You just keep building. They're like little factory workers building away. And it's amazing that we can do this in a shutdown because of a pandemic. You're doing this all the time. And then once it's done, the little ribosome falls off. Here's the stop codon. So it says that's the stopping point. It falls off, and then this is going to fold up into a protein. Okay, so that was section 5. Take a breath. Obviously, stop it whenever you need to and come back later. And this section is on gene regulation and how the cell can actually like regulate genes and turn them on and off. And we will focus on prokaryotes, and eukaryotes are a little bit more complicated. So prokaryotes are simple, right? And we're going to look at a model, I think, with E. coli. I think that's the model that we'll look at. And I am actually going to skip this slide. You can look at that later, but I'm going to show you the picture version if it'll let me. Okay, and I'll explain this. And this is really the level that we need to study. And if you take AP Bio, we'll get way more into the nitty-gritty, and it's really interesting and fun. But if you understand this level, you'll be fine. So here is a DNA strand. And we know the first step is to make RNA with transcription. So we're going to transcribe the DNA into RNA. But there are off and on switches to prevent this, to turn it on or turn it off, okay? So in this first scenario, this is a scenario without lactose. Lactose is a sugar that we find in milk, right? And sometimes people are allergic to it, allergic to lactose, because they're lacking this gene. They're not able to do what I'm going to show you. And so what this gene does, this is a gene for LAC, L-A-C, and that is a gene that makes lactase. Let me write it. LAC, and that's in italics, is a gene that makes lactase. Now, lactase is an enzyme that breaks down lactose. Let me see if that shows up for you. Hopefully it does. So lactase is a protein that's going to break down lactose. And if you don't have that gene LAC, then you're not able to make that enzyme, and you can't break down lactose, and it makes you really flatulent and have your tummy hurts and lots of problems. And you can take lactase. You can actually take that enzyme as a pill, or just not drink milk, right? So here's the gene, that's LAC. It's going to make lactase. But if you don't have any lactose, if you didn't drink any milk, you don't need to waste time making lactase. You don't need the enzyme. You don't have any lactose to break down, right? So it would make sense to turn that gene off. And here's how this is an E. coli, but this is how a prokaryote would turn off that gene. It would have this little repressor here in this region, which is called an operon. So this whole red part is an operon, and it's made up of a promoter and an operator. And a repressor is going to bind to that operator, and then here's your RNA polymerase that would be making RNA, and it's going to be blocked. It cannot make RNA. It cannot be transcribed. It's blocked. So that's the off switch. Now, if you have lactose, look at how cool this is. I think this is marvelous. If you have lactose, the repressor really likes lactose. That's what it bonds with. So if you drink milk, the repressor is actually going to fall off and bond with the lactose. The lactose is going to keep it busy. And then look at our RNA polymerase. Our RNA polymerase is able to zip around. Nothing is blocking it, and it can make RNA. So it's going to zip, do some transcription, make RNA, and then that next step is going to be, it's going to translate into protein, which is lactase. So this is an example of how a prokaryote has on and off switches. And I think it's pretty fantastic. There's a lot of variations of this in prokaryotes, lots of different versions and types, but they all kind of follow that same logic. I think they're super logical. Now, eukaryotes are more complicated. I'm not going to dig into this very much, but you can see that the gene, the DNA is this blue part. It also has a little promoter region. That's the on and off switch. Stuff has to bind to it, and then it loops around. You make a little loop, and when that touches, then that allows the RNA polymerase to go transcribe that gene, and then it's going to be translated into protein. So that's how eukaryotes turn genes on and off. And that's called a TATA box right here, because it's made of a TA on your DNA sequence. And then there's also ways where we can slow down transcription, and we can also speed it up. So it's like you can really fine tune it. Your cells are really sophisticated, and they're able to turn these on and off really well and do some cool stuff. So, okay, so after we've done, this is in eukaryotes. After we've made the RNA and we've done transcription, then the next step is to edit it, and that's called processing. So we're going to do RNA processing. We are still in the nucleus, and to do that, we cut out pieces. So we add a cap and a tail to protect the ends of our little RNA sequence. And then we cut out the introns. So you can see here those introns, those are actually removed, and we keep the exons. So the exons are the parts that this whole chunk right here is going to leave the nucleus and get translated. So I remember this by the exons exit the nucleus and they're going to get translated. And here's the 3 steps that I just showed you. Introns are removed, they stay in the nucleus. Exons are spliced together. We're going to use them, and then we add a cap and a tail. Okay, so that was section 6. Section 7 finally is mutations. So how can some things go wrong and how can we form mutations? And so some mutations can affect just a single gene, and some can affect a whole chromosome. If you look at the poor devil's kind of extra credit thing, you'll see that sometimes mutations can be a whole chromosome. And I hope you look at that. It's good stuff. I spent some time on it, and now we can't use it. So we can have different types of mutations. A point mutation is like the most minor, and that's when we substitute one nucleotide for another. Oh boy, where am I going to put that? So here's an example of that. This would be like the normal sequence. So here's a piece of DNA, that's our reading frame. This is the RNA that would bond with it during transcription. And then this is the protein that would be made from it during translation. And then if we just switch out this one guy right here, I think, am I in the same place? So the CG is being switched out with an AU over here, just one nucleotide. This is a point mutation. It's a substitution, like a substitute teacher. You're switching one in for another. It's going to change that amino acid. So it's going to make an asparagine instead of a glutamine. So that's an example of a mutation. And then we can also have frame shift mutations, and that's where something is getting added in and shifting everything over, or it's getting deleted and shifting everything over. So here's the normal sequence, and then here we're adding in a G, or no, we're deleting a G. So we had 2 Gs here, now we only have 1, and it's going to shift all the amino acids that come after that because it's shifting everything over. So frame shift are pretty bad, and then point mutations aren't as bad. And this could happen with SARS-CoV-2 and has happened. We're seeing some changes in the nucleotide, some small changes, and even some different strains. And that often happens with viruses. One note though, this is just me speculating, but I'm hoping that since we have most people have mild symptoms, 80%, I wish it was higher, and those are being transmitted a lot. There's a high rate of transmission of the people with mild symptoms. I'm hoping that if there were mutations going on, they would be with those populations, and maybe they would shift the virus to be less harmful. But that's just me speculating. I don't know if it actually works like that. I'm sure that is known. So here's another example of a type of mutation. A chromosomal mutation can happen when, during crossing over, we cut the wrong pieces of chromosome and we lose genes. So sometimes we make duplications of genes or we delete genes during crossing over. So that can also be problematic. And then we can also do translocations where we have chromosomes that cross over that are not matches. They're not homologous chromosomes, and we can get weird chunks crossing over. Okay, so the chromosomal mutations can have big effects because we're like cutting and pasting whole genes. Bad stuff can happen. We can also have some bad things happen with gene mutations. So those are like single nucleotides. We can have a premature stop codon that'll stop translation from happening. It can change the shape of the protein, and it can also change gene regulation. So those operons, those switches are not turning on or off when they're supposed to. Okay. And some mutations are silent. So sometimes they don't affect anything. Sometimes they don't change the amino acid sequence. since we have, like, remember for leucine, there were 6 different codons that could code for leucine. Sometimes it just mutates to another one of those codons and it ends up coding for the same amino acid. So that can happen too. Also, it could happen that the mutation is in a region that isn't even a gene. A lot of our DNA doesn't really do anything. It's probably from our ancestors. Or it could be in a part that it doesn't really affect how the protein folds. The protein is still going to fold okay and be normal. If you have a mutation in your body cells, like if I have a mutation in my hand, that's not going to affect my offspring, right? Only the mutations in our sex cells are going to affect our offspring, because that's what the offspring are actually made of, that genome. And also, if it's a harmful mutation, then sometimes natural selection will kind of select for that, because maybe it'll code for an adaptation that's not so good. Maybe that'll happen with COVID-19, and that won't be passed down. Or sometimes it's a good mutation, and then that will be passed down through natural selection. Okay. Now, mutations, we've talked about what could cause them because we looked at cancer, but obviously mutagens like UV radiation and chemicals and carcinogens can cause mutations. And sometimes cancer drugs do it because they're trying to kill cancer cells. So those are some things that can definitely cause mutations, but sometimes they're just random errors that happen when your cells are replicating their DNA. Okay, so I'm going to switch over, stay with me, to the version that has the questions, and we'll just go through them, and I'll read them, and then you can just answer them on a piece of paper to yourself. We're not taking a grade, but if you would like to kind of quiz yourself, you'll have this also available to you. So first, we'll start with section 1. Which results of Frederick Griffith's experiments led him to believe in a transforming principle? And I'm going to go quickly. I would like you to just pause. So I'll give you a little gap in time so I'm not rushing. But you can pause and write down your answer and then check yourself. Okay, so that's that. An important factor in Avery Oswald's ability to identify the transforming principle as DNA was the fact that proteins what? So how did Oswald Avery know that DNA was the transforming principle? This was the second experiment, following up from Griffith's experiment with mice. Okay. Contained very little phosphorus. Chemical analysis of Avery's extract showed that the ratio of nitrogen to phosphorus was consistent with that of what? So what did he find it was consistent with? Okay, there's that. Hope I'm not going too fast. Hershey and Chase chose to use bacteriophages, which are viruses that infect bacteria, in their experiments because these viruses, I hope I didn't give away the answers. No, I didn't. Okay. And as a result of Hershey and Chase's experiments, scientists believe that what? So this is the third set of experiments. What did they finally conclude from this? All right, so I'm going to skip ahead to the questions at the end of section 2. Oh, 4 types of nucleotides that make up DNA are named for their what? And if we were doing a bonus here, I would ask you, what are those 4? And those are, oh wait, what are those 4 nucleotides? They are A, T, C, and G. And then you can think about which ones are pyrimidine single ring and which ones are purine double ring. So the C and the T are pyrimidine single ring, and then the A and the G are purines double ring, and then U is also a single ring pyrimidine. After examining the DNA of different organisms, which of the following did Erwin Chargaff conclude about the 4 bases? This one's a little tricky. 2 of the answers kind of seem the same, but that is A. So we're showing here that A's bond with T's and C's bond with G's, and not necessarily, they don't add or equal. So that's why A is better. Which of the following DNA sequences is complementary to the base sequence ACCGTAT? So we're looking at which one would bond with it. So look at which would bond with the A and start with that. Okay, so that should be the match. And you're never going to see a U and a T in the same sequence. U is in RNA and T is in DNA. So that one is a what? That one doesn't make sense. Combining the work of other scientists with their own research, Watson and Crick discovered that 2 strands of DNA joined together to form what? And so we call that a double helix. So that's that twisted ladder. And then what holds those base pairs together in the middle? What holds the rungs together? This is really important for how we do replication and transcription. So those are hydrogen bonds, and those are really weak bonds that can easily unzip. Okay, now section 3's questions. The process of making an exact copy of the cell's DNA. So what is that process called? That is replication. And then you can ask yourself, when does that happen during the cell cycle? And we know that it happens during synthesis when it's getting ready for mitosis. What are the 4 functions of DNA polymerase? No, that doesn't say that. What are the main functions of DNA polymerase? Okay, so that's bringing in new nucleotides and then making sure that they're correct. Which of the following events occurs directly after the DNA molecule is unzipped? And remember, it's unzipped by helicase, that enzyme helicase. So what happens after the DNA molecule is unzipped? So the free-floating nucleotides come in and bond, and the DNA polymerase is facilitating that. The process of making new DNA molecules is semi-conservative. This means that every new DNA molecule is composed of what? So we're copying the DNA. What are the new molecules made of? So we have 1 original and 1 new strand. When DNA molecules are formed, almost all errors are detected and fixed by what? So what's finding those errors and fixing them? It's proofreading and revising. Okay, so that is DNA polymerase. All right, now we're on to section 4. Woo. Okay, the central dogma of molecular biology states that information flows in one direction from what to what. This is our big picture of what's going on in this process. And that is DNA goes to RNA through transcription and then goes to proteins through translation. Choose the nucleotide sequence of RNA that would be complementary to the following DNA strand. So what would match that? And remember, RNA has U's instead of T's. So the answer is Kaukagoo. So we have CAUCAGU. Okay, the main function of tRNA is to do what? So we're bringing amino acids from the cytoplasm to the ribosomes and matching them. Which of the following events occurs directly after RNA polymerase recognizes the transcription start site of a gene? So the DNA strand unwinds and it separates the 2 strands. And then the primary difference between transcription and replication is that transcription does what? This is a good question. That is A. Transcription happens a bunch of times. Replication only needs to happen once because then the cell splits. So transcription, you can transform lots of genes over and over and over. Okay, now section 5. Okay, what is the term for a 3-nucleotide sequence that codes for an amino acid? So we had that big table of 3 nucleotides, and then each one matched different amino acids, and those are called, that's a codon. Okay, how many amino acids are used to make up all the proteins in the human body? How many different amino acids were on that table? I gave you that number. They're throwing in here chromosome numbers to confuse you. So that answers 20. 23 is how many different chromosomes we have, and then 46 is your pairs. So 23 is the pairs, actually. Which phrase best describes translation? So that is A, converting mRNA into a polypeptide. Which of the following is the site of translation? Where does it happen? That's what we're asking here. Where does translation happen? Where is the protein made? So that is ribosomes. Okay, section 6. Okay, oops, whoa, sorry. In prokaryotes, gene expression is regulated by controlling what? So you saw the answer. We're controlling transcription, and we have that on and off switch on the DNA in front of the gene that can either turn on transcription or turn it off. The bacterial lac operon is switched on when what happens? When do we turn that lac gene on to make lactase? We turn it on when there's lactose present and it binds to that repressor molecule. In a eukaryotic cell, where does mRNA processing take place? Where are we processing that mRNA and cutting parts out and putting the cap and tail on it? We're still in the nucleus when we do that. We're getting it all ready to leave. What is the term for the nucleotide sequences that are removed during mRNA processing? So the chunks that are taken out and trashed are the introns, okay? The exons are the parts that we use. And what is the main function of a promoter? So those promoters tell that RNA polymerase where to start, and they can turn genes on and off. All right, last section, almost done. Okay, generally mutations that affect a single gene occur during what? When do these mistakes occur? If it's just a single gene. So that's usually going to happen in replication, and we make a mistake that the DNA polymerase doesn't catch. Cystic fibrosis is an example of a genetic disorder caused by the deletion of a nucleotide. What is this type of mutation called? If we have a deletion and then everything scoots over, that is a frame shift. Which type of mutation has no effect on phenotype? It doesn't affect the amino acids that are being made. That would be a silent one. Mutations that can affect the offspring of an organism occur in what cell type? So where are the mutations actually going to be passed down? And that would be in gametes. So that would be in your sex cells. And then which of the following is an example of a mutagen? Which is a substance that can, or something that can cause a mutation. So it would be sunlight. Sunlight can cause a mutation. Okay, so that's all of it. And remember that we have Zoom scheduled for each day at 1 if you need help. And let me know if you have other questions or if I can help. See you guys later.